Good evening, students. How are you all? कैसे हैं आप सभी? I hope you all are fine. So, before going to start today's topic, I wish to start with a quote. If you want to win tomorrow, you must start it today. It's mean you should be give your best at every moment. इसका एक सिंपल सा उदाहरण है. अगर हमने छत पे जाना हो तो क्या ऐसा हो सकता है कि हम पहली सीढ़ी पे पैर रखें और सीधा फिर ऊपर छत पे पहुंच जाएं? ऐसा नहीं हो सकता. हमें स्टेप बाय स्टेप वी नीड टू फॉलो each and every step, and then only we can reach the top of the roof. Similarly, if you want to be successful in your examination, definitely you have to start preparing yourself from today only. From today only you have to start preparation. So in this sequence, today we will talk about a very interesting topic in cell cycle, that is meiosis. We have already discussed in our previous video that is related to mitosis. Now we will talk about meiosis. The production of offsprings by the sexual reproduction include the fusion of two gametes. We know that male gametes and female gametes, and fusion of male and female gamete resulting in the formation of zygote, and that will lead to the development of new organism. So gametes are formed from the special diploid cell. The cell division that reduces the number of chromosomes into half and result in the production of haploid daughter cell is called meiosis. So meiosis is very important. It restores the same number of chromosomes in the member of species. जैसा कि आपको पता है हमारे पास कितने क्रोमोसोम होते हैं? 46. So out of 46, whenever this meiosis division will takes place in the germ cell, when gamete formation will takes place, 23 chromosomes will go to gametes and 23 remains with the parents. Similarly, the 23 chromosomes from the father and 23 chromosomes from the mother. So there are 46 chromosomes in the offsprings in human beings. So meiosis involves the two sequential cycles of nuclear and cell division. एक nuclear division होती है, एक cell division होती है, जिसको हम meiosis 1 और meiosis 2 कहते हैं, लेकिन single cycle of DNA replication होती है. It involves the pairing of homologous chromosomes and recombinations of them. 4 haploid cells are formed in this process. In mitosis, we have studied that 2 daughter cells are formed. Here we will talk about 4 daughter cells are formed. Then this is divided into, for example, when we are talking about prophase 1, it is divided into leptotene, zygotene, pachytene, diplotene and dikinesis. So when we talk about leptotene, during the leptotene what will happen? The chromosomes become distinct and visible under the microscopes. The compaction of the chromosomes continued throughout the leptotene phase. यहाँ पे chromosomes जो हैं वो काफ़ी compact हो जाते हैं और आसानी से आप उनको देख सकते हैं, visible हो जाते हैं. नेक्स्ट स्टेज इज जाइगोटीन. क्रोमोसोम स्टार्ट पेयरिंग टुगेदर. आपस में क्रोमोसोम मिलना शुरू हो जाते हैं और वो एक स्ट्रक्चर बनाते हैं जिसको कहते हैं सिनेप्सिस. द पेयर्ड क्रोमोसोम स्टार्ट पेयरिंग टुगेदर called homologous chromosome, and they will also form synaptonymal complex by a pair of homologous chromosome. This is called as bivalent or tetrad. This is a very important phenomena during this stage, zygotene. The first two stages of prophase 1 are short lived as compared to next stage, that is pachytene. So when we talk about this pachytene stage, the crossing over between non-sister chromatid of homologous chromosomes occur for the exchange of material, and this crossing over result in the recombination. We got lot of variations. We say that sexual reproduction leads to variation, and this variation, the root cause behind the variation is this division, meiosis. And during the meiosis, what we are saying, that there is a exchange of genetic material, that is crossing over, and there is an enzyme which involved in this process. You can see here in this slide, the enzyme is recombinase. It leads to the recombination of genetic material of 2 chromosomes. Then we have diplotene. It is recognized by the dissolution of synaptonymal complex. जो synaptonymal complex बना था वो separate हो जाएगा, dissolve हो जाएगा. Tendency to separation of bivalent except at the site of crossing over. सिर्फ crossing over पे वो जुड़े रहेंगे, बाकी जगह से बढ़ जाएंगे, and that form a X-shaped structure. This is called chiasmata. In oocytes, some vertebrate, diplotene can last for months or years. We know that during the menstrual cycle, what will happen? This oocytes, actually ovum become mature, and these follicle cells remain in the female's ovaries from the time of birth, and when they reaches the puberty, then they will become mature and they will form the ovum. So this thing we have to learn, that in oocytes of some vertebrate, diplotin can last for months or years. Diakinesis is marked by terminalization of chiasmata. The nuclear membrane breaks and nucleolus disappear. Then we come to another phase, second, that is metaphase 1. Bivalent chromosomes align at the equatorial plate. They will align themselves at equatorial plate. Microtubules from the poles of the spindle get attached to the pair of homologous chromosome. माइक्रोट्यूब्यूल जो हैं वो पोल से स्पिंडल्स के साथ उनको अटैच करेंगे. दिस इज अबाउट द मेटाफेज 1. Then we have anaphase. So what will happen in this actually metaphase? Poles of the spindle get attached to the pair of homologous chromosome. Then anaphase 1, homologous chromosome get separate but sister chromatid remain attached at the centromere. Sister chromatid जो होंगे वो centromere में attach रहेंगे. Telophase, the nuclear membrane and nucleolus reappear. Cytokinesis follows. This is called as diode of the cell. The stage between two meiotic division is called interkinesis. दो meiotic division के बीच की जो stage होती है उसको interkinesis कहते हैं. Short lived होती है and that follows the prophase 2. और उसके बाद कौन सा फेस आएगा हमारे पास? प्रोफेस 2. तो जैसे कि मैंने कहा, प्रोफेस 2, meiosis 2 is initiated immediately after the cytokinesis before the chromosomes have fully elongated. यहाँ पे chromosomes पूरी तरह elongate हो जाएंगे. In contrast to meiosis 1 and meiosis 2 resembles normal mitosis. जो meiosis 2 है वो बिल्कुल mitosis की तरह ही है. The nuclear membrane disappears by the end of prophase 2. The chromosomes become compact. Chromosomes यहाँ पे compact हो जाएंगे. जैसे metaphase 2 आएगा, chromosome align at the equator and microtubules attach with the kinetochores of sister chromatid. Kinetochores is attachment where chromatids will attach. जो arms होती हैं chromosomes की वहाँ पे attach होती हैं. Anaphase 2 start with the splitting of centromere of each chromosomes to move toward the opposite pole. Meiosis and with telophase 2, in which the two group of chromosomes get enclosed by nuclear membrane. इनके बाहर nuclear membrane लग जाती है और cytokinesis होता है and it form tetad of cells. 4 haploid daughter cells. As I have mentioned that in meiosis 4 haploid daughter cells are formed. So what is the significance of meiosis? That is another important question. What is the significance of meiosis? So when we talk about this meiosis 2, we have already discussed that what will happen in prophase 2. That in prophase 2 initiated immediately after the cytokinesis, usually before the chromosomes have fully elongated, and in contrast to meiosis 1, meiosis 2 resembles the mitosis division. Then metaphase 2 में क्या होगा? Chromosome align at the equator. Anaphase 2 में क्या होगा? Start splitting of centromere. Each chromosomes move toward the opposite poles. Then telophase 2, it will form into 4 daughter cells. So that we have to learn. Then what is the significance of meiosis? Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved. As I have mentioned that we have 46 number of chromosomes. So during the reductional division, during the spermatogenesis and oogenesis in female, what will happen? This chromosome numbers reduce to half. And when the fertilization will take place, zygote is formed. Again that chromosome number restores to the 46. Then it increases the genetic variability. We know that during the sexual reproduction there is a chances of variation, and variation are very important for the survival of organisms in the changing environment. Variation helps in adaptation. Variation जो होती हैं organism को adaptation में help करती हैं. And variations leads to evolution, which is very important phenomena and very interesting phenomena. Evolution. Small gradual changes which takes place in an organisms and they accumulate in the form of a larger changes. So this evolution is also very important for the survival of the species in the changing environment. Then we talk about this diagram. Mitosis we have already discussed, and for the diagram of meiosis you can simply see here. This is the diagram. Prophase 1, metaphase 1, anaphase 1 and telophase 1. You can see here 2 chromosomes are formed. Then prophase 2, you can see here prophase 1, metaphase 1, anaphase 1 and telophase 1. And then similarly prophase 2 leads to 2, and then ultimately it leads to the formation of 4 daughter cells. So this is about meiosis. This is a very simple process. I hope this is clear to you. So what we have studied, that meiosis is very important phenomena, and what is the main important significance? That it leads to the recombinations. It leads to the recombination. And another very interesting thing that is related to this meiosis is that it increased the genetic variability in the population of organisms, and it also restored the same number of chromosomes in a particular species. So I hope this video is definitely going to clear all your doubts related to meiosis division. Thank you so much.